So to start off, the whole problem of how the process of axonal elongation works is very much a problem in cell motility. And to start this off, I begin with a really famous movie taken in the 1950s of a white blood cell that is migrating around, chasing a bacteria. So I'll just sort of point out a couple of things. So here's going to be a white blood cell, and any guesses on what these dark circles are?
Red blood cells.
In any case, yes, indeed, these are red blood cells. So more or less, this is just a blood sample with some red blood cells here. We have a white blood cell up here. And then there's some little black dots that are skating around that are bacteria. And one thing that I think is really remarkable, so here's a bacteria here, and you've got your cell, is that it really looks like the cell is hunting down the bacteria. That the cell has some sort of intelligence in a sense, that it can sense the environment, it can respond to the environment, it can chase down prey, and respond in very appropriate ways. In terms of how this happens, there's two sort of major regions of the cell that we're going to talk about. We've got the lamellipodia at the very front, and well, actually, I'll just point out. So, do you notice at the very leading edge of the cell, it's relatively clear, and it looks really sort of lumpy in the middle? So, what we have here is where the organelles, the cell body is going to be contained with the nucleus of the cell. And at the very front, we have a region called the lamellipodium, which is very rich in actin. And what the lamellipodium does is it grabs onto the substrate and it generates these forces that help drive the process of cell migration. And at the very back, we have this little tail region as well that's just kind of being dragged along. With that sort of background, what we want to understand is the cytoskeletal proteins that in fact control and drive this sort of process here. And there's three main types of cytoskeletal elements in cells. There's microtubules, there's actin filaments, and there's intermediate filaments. It turns out that actin filaments are very often called microfilaments. And when we look at the cell, we have the leading edge of the front of the cell right here. Here's the nucleus of the cell. There's microtubules in the center, and there's intermediate filaments as well. So when cells migrate in this direction, and what they're doing is they're interacting with the substrate at the front. And I'll just pause for a second and ask if there's any questions at this point. Okay, all right. So getting into one critical idea in cell biology is that of polarity. So things have direction. And this is an electron micrograph of microvilli in an intestinal epithelial cell. So inside our gut, there's a layer of epithelium, and the cells that project in the gut have these filopodia. And I'll just pause for a second, and what's one of the major things that needs to happen in the gut, in your digestive tract?
Like the absorption of nutrients?
Exactly. And so in terms of solving this problem of nutrient absorption, one sort of thought is that in the gut, the microvilli greatly expand the surface area of the cell. So you have all of these sort of extensions that are sort of reaching out, and that maximizes the surface area of the cell and also the intake of nutrients. And in the lumen of the gut, nutrients are going to be absorbed, and then material needs to be passed through. So in terms of the cell itself, there's a polarity. So there's an apical domain, or the top of the cell, that is in the lumen of the intestine. And then there's a basolateral domain, or a back of a cell. So cells have a front and a back. They have it when they migrate. They have it when they're in tissues. And the underlying basis of this polarity is that the cytoskeletal elements that build up a cell also are polar structures. The molecules themselves have a front and a back. And we're going to talk about that now. So one way to think about the cytoskeletal elements in cells is to make an analogy with our body. So the microfilaments in actin, along with myosin itself, you can sort of think of as the muscles of the cells. It generates contractile forces. So when cells are grabbing onto the substrate and pulling back, they're using force generated by myosin to do that. So actin is kind of like the muscle in a cell. Microtubules are long, hollow cylinders that can handle compressive forces. And you can kind of think of them as the bones inside of the cell. So very often cells need to sort of put out long extensions. So for example, in cilia and flagella, there's long microtubules. So the microtubules are important for that. And they also serve for railroad tracks. And the intermediate filaments, which are called neurofilaments and neurons, are more sort of like tendons or connective sort of tissue. They don't generate forces by themselves. They're not important for transport. They're just holding things together. And as one little tidbit, microtubules and actin are incredibly important for understanding how neurons grow. Neurofilaments less so. It turns out that in invertebrates, for example, like fruit flies, they grow neurons just fine, but their neurons don't have intermediate filaments, neurofilaments like vertebrates do. So we won't talk so much about intermediate filaments in the context of axonal elongation, just because there's not a lot of evidence that it's the central part of that process. And as just one final point, these are 3 images of the same cell. So this is labeling up the actin filaments, the microtubules, and the intermediate filaments. So these sort of short lines are here. Those would be actin stress fibers. Each individual sort of line here is a microtubule, and then you can kind of get a sense for how the intermediate filaments are found in cells. So to talk a little bit more in depth, next I want to talk more about actin and how it is built up. So while microtubules are built up of the protein tubulin, actin is built up of polymers of the protein actin or actin filaments. And actin forms these filaments here. Actin is very often found right against the plasma membrane. It serves as a track for myosin for contractile force generation, as I had sort of mentioned, and also interactions between actin and the outside world through molecules like integrins are important for force generation. Microtubules, polymers of the protein tubulin, once again, structural framework, tracks for transport, and in particular, the molecular motors kinesin and dynein use microtubules to transport organelles and proteins. So in neurons, 99% of proteins are made in neuronal cell bodies, and those cell bodies are in our spinal cord. And then we have neurons that reach out for a distance of 1 meter. So the microtubules are the tracks for that transport of material from the neuronal cell body out to synapses and distal corticals. And then finally, intermediate filaments, polymers of intermediate filament proteins, and once again, structural support, but there are no known motors that are involved with intermediate filaments. So, okay. So that's kind of a quick overview of the different cytoskeletal elements, and I'm guessing you're probably all familiar with this sort of stuff. This is pretty basic background material, so okay. One of the sort of ideas or things I wanted to show you with that movie where there's a leukocyte, a white blood cell chasing down a bacteria, is that cells can respond to the environment. And very often, or at least in that particular case, the sort of thought is that the bacteria are releasing molecules that the cells can sense, and when there's a gradient in the concentration of those molecules, the cell can change where actin's being assembled, where myosin is contracting in order to make the cell move in that direction. And we get this sort of connection. So on the outside or the exterior of the cell, there's going to be signals. There's going to be receptors in the membrane that are going to be able to sense those signals. And then there's signal transduction pathways that control the cytoskeleton to control cell shape and movement, as well as the transport of materials from one region to the other. So in the end, we want to understand how cells sense the world, how they respond or process that information, and then generate the forces to actually do things in the real world. We've got a ribbon diagram here of actin, and we can tell it's a protein based because we're looking at a ribbon diagram. As we know, at the beginning of all proteins, we have the N-terminus, and then at the very end, there's the C-terminus. When we look at this ribbon diagram, we can see that there's these coils here. Does anybody remember what the coils are called in a protein?
Alpha helices?
Very good. So those are alpha helices, and then we also have some other structures that are lined up like this. So what are these called? Yeah, so these are the alpha helices and the beta sheets that make up the secondary structure of the protein. Very often, so what happens in the beta sheets is that there's hydrogen bonds that are letting individual atoms interact with each other. And when proteins need regions that are strongly and tightly held together, very often you see beta sheets here. And if we can think of actin as sort of this brick or this building block in cells that experience really high forces, you can imagine that the internal structure is going to be very tightly held together and compact. Actin is a really highly conserved protein. So human and amoeba actin are 80% identical despite a billion years of evolution. And one way to think about that is actin is an essential protein for a lot of different processes like cell division, cell migration. And when things are really critical and important, if one tiny bit of it gets changed, then that can disrupt multiple processes. So there's a lot of selection pressure against that sort of thing, especially for these essential highly conserved proteins. Okay. So if we look at the structure of G-actin, it's about 5 nanometers in diameter. And if we go back to our sense of scale, so we can think of individual atoms as about 1/5 of an inch or about the size of like a green pea or something like that. Proteins themselves are kind of the size of, say, like a basketball, and things like a mitochondria would be the size of like a football field. So actin is like a pretty, reasonable sized protein. And then we've got this molecule in the middle, ATP. We know that ATP is really important for energy, which we've talked about. And it turns out that the assembly and disassembly of actin is regulated by ATP hydrolysis. So ATP gets hydrolyzed during the process of actin assembly, and it's a critical part of controlling both assembly and disassembly. And we'll get into those details in a bit later on. Okay, so like so many things in biology, actin goes by different names. So an actin filament, I very often call an actin filament, but sometimes people just might call it actin, or F-actin, or microfilament. These are things that all mean the same thing. And then just in terms of the subunits themselves, we have things called, the subunits are soluble. So very often that would be called soluble actin, globular actin, because they make these little globes, abbreviated very often G-actin and monomeric actin. So they all mean the same thing. So The subunits, these are different names for the subunits. The filaments, these are different names for the filaments, and they get used pretty interchangeably. So actin filaments appear as two strands arranged in a helix. So if we've got our actin filament here, as I mentioned, cells are polar structures. They have a front and a back. Actin filaments are polar structures. They have a plus end, they have a minus end. And when we look at the subunits themselves, we also have a polarity. And if you sort of focus in here, what you'll notice is that in the cartoon, there's a little dimple or a little sort of cleft in each one of those subunits. And if we go back here, that's what that's trying to show. So you can sort of think of actin as a protein that has a little cup on the inside, and that ATP is going to sit in that cleft. And when we look at the filaments, we can see that the cleft points in the same direction with the region where ATP can get in and out at the minus end, and then the sort of backside at the very front here. Okay. Okay. So actin goes through some basic chemistry where there's soluble subunits that then get polymerized to form these filaments. If we start off with a solution of soluble or globular or G-actin here, and let's say we have a really high concentration of soluble actin in solution, spontaneously, just by chance, there's going to be nucleation events where 3 monomers come together and they form that short little polymer. And then after that polymer is formed, there's going to be elongation. And if the concentration of actin is really high, there can be elongation on both ends of the filaments. If we start off with a solution of soluble actin and let these actin filaments assemble, over time, the concentration of soluble actin is going to decrease, the concentration of polymer is going to increase, and you're going to reach a steady state situation where the addition of subunits is balanced by the loss of the subunits, and the concentration of filaments is steady over time. Let's see here. And it turns out that the concentration of G-actin when things are steady or at steady state is called the critical concentration. So if you start off with actin, let it go to steady state, there'll always be the same critical concentration of actin. If the actual concentration is above the critical concentration, you're going to get net assembly. If it's below the critical concentration, there's going to be disassembly. So, if we look at a graph of actin polymerization over time, you can see we've got the mass of actin filaments, and so once again, we're starting off with just a solution of soluble actin. We put it in a test tube, and we can actually use light to measure the amount of actin filaments. So what happens is when actin assembles, it makes solutions look cloudy, which blocks light. So essentially you can take a tube of actin, shine light through it, and then based on how much light is transmitted, you can measure indirectly polymer mass. What you can see is that, at least in this case, initially there's kind of a very slow rate of mass gain, and then it really rapidly increases, and then it flattens out over time. And I had mentioned that the nuclei are an important step in actin assembly. So we've got our little nuclei here with the 3 actin subunits, and the question here is how can we test that the formation of nuclei are an important step in actin assembly? So we'll just kind of take a little break here and talk about how we would do like this experiment. So how do you test anything? So the hypothesis is that formation of nuclei are important for actin assembly. So we want to say the nuclei are doing something. If you want to test anything, like if you're saying, I don't know, bees do something, how would you test if bees do something? Exactly, yes, yes, exactly. Okay, so we want to see if the nuclei are important. How would we test if the nuclei are important? Yeah, go ahead.
Maybe have one without a nuclei and one with a nuclei and see what is different.
Exactly, yeah. So like the heart of any experiment is you want to know if something's important. So you add that something, and then you also have a control where that something's not there, and you see if anything's different between those cases. So one way to make actin nuclei is if you've got a bunch of F-actin, you can just take it up into a syringe and go back and forth a bunch of times. And what that does is it breaks the filaments apart into these short little fragments. So we can start off, we can say this is our control. So we can measure the mass of actin filaments, and we know that initially there's a slow sort of assembly and then it takes off. Another thing that we could do is add these nuclei at the beginning and see what happens. And I'll just show you the results. This is what happens. So if you start off nuclei added times 0, you get boom. Well, actually, I'll just say, what happens when you when you do this?
The elongation state is immediately.
Exactly.
The addition.
So, and what does that imply?
The formation of it. Like it's forming longer.
Pardon?
It's getting bigger.
It's getting, yeah. So what is it? So the fact that we see essentially really slow assembly and then rapid, but if we have the nuclei, it just takes off. What that really sort of is getting at is that it takes time for these nuclei to form in solution. And initially, there's just going to be a few filaments that are forming. And then as more nuclei form over time, elongation really takes off. If you just add the nuclei here, there's going to be lots of actin filament ends that then new growth can take off of. So you get this really rapid response. And it turns out cells don't want or need actin assembly everywhere. And there's a lot of mechanisms that are actually preventing actin from assembling everywhere in the cell. And one of these mechanisms is that nucleation is an improbable event. So this is something that happens infrequently. It is part of what regulates this whole process of actin assembly. So, okay. All right. We mentioned that actin is a polar molecule. And one of the experiments that first really demonstrated that was to take myosin, fragments of myosin, and mix those with actin filaments, and then look under electron microscopes at the structure here. And I'll just go ahead and tell you, so this is an EM, here's an actin filament, and the head region of myosin, which binds to actin, has been added. And when people first looked at pictures like these, they noticed these sort of repeating structures that kind of look like arrowheads. And there's kind of a pointed end here, and then you've got a barbed end on the other side here. So does everybody sort of see the repeating sort of belt? Okay. And it turns out you would think that the pointed end would be the plus end or the front, but with the actin filaments, it's the other way around. So it turns out that the barbed end, when labeling with myosin, is actually going to be the plus end, and then the pointed end is going to be the minus end. And this barbed and pointed end isn't used in the literature so much now, but historically it was used a lot and stuff like that. Okay. So we built up this idea. So we have this assay that we can look under an electron microscope, and we can sort of tell the direction that an actin filament is pointy. We have assays where we can add soluble actin and form filaments and whatnot. And now the question is, how can we test if the dynamics of actin assembly are the same on both ends with the tools that I've just sort of talked about and discussed? Go ahead.
You would put solute in the solution and measure from, like, if one side grows faster and one is shorter.
That is an elegant solution to that problem, and this is the result of that experiment. And there's a certain beauty of that. It's a simple idea. Yeah, we can label up the filaments. We know what is the myosin. We know what is the plus. You throw soluble actin in, and you just look and see. And it's really quite clear. So the plus end, there's very rapid assembly, and then on the minus end, it's a lot slower and stuff like that. So very nice. So we've got that. Now, this is just a static picture. What would be a nicer way to see if the plus and the minus ends had different dynamics and actually maybe measure growth rates? You take a movie, and this is that movie. So in this case, I think they used like rhodamine actin. So they fluorescently labeled the soluble actin, and then they did experiments. So these are individual actin filaments. And can you tell which one is the plus end? Is it on the left or the right? Yeah. And when you see it in the movie, it's really obvious and stuff. And we essentially have like the technology to do experiments like this and like take movies like this. So, okay. And then, So to really understand these dynamics, the next thing that we need to do is to talk about the equations that describe actin assembly and disassembly and whatnot. Okay, all right. So the next thing we want to do is we want to talk about why the rates of polymerization at the plus or minus ends are different, or at least describe the sort of like, biology that underlies that. I guess the way to start off is we need to talk about ATP hydrolysis along the length of these actin filaments. It turns out that soluble actin, mostly like 90+% binds to ATP. And when actin is assembling on the plus end, it turns out that initially there's going to be ATP bound to actin. Over time, that gets hydrolyzed, so there is now ADP plus an phosphate in the actin. And then eventually what happens is as the actin ages, that phosphate gets released, and then it's just ADP itself that's there. And that PI, that inorganic phosphate, is coming out of the cleft here. So over time, ATP actin gets hydrolyzed to ADP actin. And as we know, phosphate groups are very large molecules. There's typically like 4 oxygens that are surrounding the phosphorus. And when that phosphate group is lost, it changes the shape of the actin itself, just because now those minus signs, those are negative charges, they're not repulsing things, so then the actin can change conformation, and when the actin changes conformation, it does different things. It turns out that change in conformation, in part, also changes the way that the ends interact with soluble actin. And the way to think about it is when actin first assembles, it has a full ATP, and a new subunit comes on, and it has the perfect shape for the addition of the next subunit. So stuff stacks up really rapidly. At the minus end of the filament, that phosphate's been lost. The actin has changed its conformation a little bit. And when it changes its conformation a little bit, it just means that it's a little bit less likely to add on that minus end. So it changes the dynamics. So this change in shape, a change in dynamics. And okay, yeah. All right. So the ends of actin filaments are different, and one way to sort of like keep track of that or really make the point is we can think of the monomers themselves as having a smooth side and a cleft side. So smooth and cleft, and you can see that carried over here and that carried over here. And this is just a different way of sort of saying that the ends of the filaments are different. They have different surfaces exposed. So, okay. So as we mentioned, the dynamics on the plus ends of actin filaments and the minus ends are different. And one of the major differences is, well, how fast they assemble as a function of G-actin concentration. Another idea that's important is that the rate of addition is a function of G-actin concentration. And one way to think about this is if you've got an actin filament and there's the soluble actin in solution, just by chance, that soluble actin has to get in the right sort of region, or that region where the end of the filament is, and it has to have the right orientation, and when all of those things are right, you get assembly. As concentration rises, the probability of that event goes up. So the rate of addition is a function of concentration. It turns out that using sort of approaches from pharmacology, by varying actin concentration and measuring rates of polymerization, we can determine rate constants. And as the first rate constant I want to talk about, so we have addition at the plus end. And what we have here is this 12 micromolar, 12 per micromolar per second. And I'm just going to pause here. So do you guys understand? So if you see an S-1 superscript, do you understand what that means? What does that mean?
A read per second. It's like a read.
Exactly. Yeah. So yeah. Okay. Yeah. So whenever there's a -1, it means like over. So this is 12 over micromolar over second. So, all right, so you get your rate constants there, and there's a couple of different rate constants here. The way to use these numbers is if we know the concentration of free actin is 1 micromolar, then what this is telling us is that, that there will be 12 actin subunits that get added per second. So these rate constants are really useful. If you know the concentration of the solution, you can know how fast things are going to grow. And then if we look over here at the minus end, we can see this rate constant is 0.3 per micromolar per second. So if we had a concentration of 0.3 micromolar, how fast would things grow? It would just, 1 per second. So yeah. Okay. And there's also the off rates. And at the minus ends, and the plus ends, the off rates are different. So when we look at the on rate, the on rate depends on concentration. If we look at the off rate, we can see, for example, on the plus end, it's like 1.4 per second. So what this is saying is the rate that the polymers disassemble doesn't depend on concentration at all. So if we think of this constant, there's only time, there's not concentration. And if we think about the reason why, so in the filament, we have our subunits like assembled into a solid. And the concentration of the soluble stuff doesn't have anything to do with that. The rate that those subunits come off of the filament is its own thing, and it just happens at a constant rate. So spontaneously, actin is going to disassemble. It doesn't matter what the concentration is, it's a fixed constant. So a lot of what cells are doing is they're controlling assembly rates, and at least in this context, disassembly doesn't matter on concentration. Cells have other mechanisms, and in fact, when cells really want to get rid of actin, they break it up into little chunks, and then that gets disassembled. So, okay. So assembly depends on concentration, disassembly does not. Okay. So in terms of bringing these two things together, I had mentioned this idea of a critical concentration, where if you take a solution of actin and let it go to steady state and measure the concentration of soluble actin, it will be constant over time. And this is the critical concentration. If the concentration if you bump up the concentration, you'll get assembly. If concentration goes down, disassembly. So when we look at this, we can see there's two different on rates and there's two different off rates. And in order to figure out the critical concentration, we need to actually calculate all of these things together. But we're going to just start with one end of the filament here. So for the plus end, the critical concentration is 0.12 micromolar. And if you sort of put these numbers together, and I'm going to explain how that gets calculated here. So to calculate the critical concentration, you just take the off rate and divide it by the on rate. And if you think about it, so we've got our 1.4 per second up here, divided by 12 per micromolar per second, that's up there. And when you put these two things together, you get 0.1 micromolar. And a lot of people have trouble just looking at the math this way in particular. And I've always found that this is a nicer way to talk about the math, at least for these sorts of things here. So is just actually, I'll do a little pop quiz. So we know molar is moles per liter. And the question I'll throw out is, what is a mole?
It's the amount of substance in 12 grams of weight. The amount of substance that contains as many elementary particles as there are in 12 grams of carbon 12.
Indeed. So that's a very precise and technical definition. Yes, yes. Yes, it is also Avogadro's number. Very, very good. No. One thing, and I don't remember if I told you this. Mole it's just a number, like a dozen stands for 12, a century is 100, a score is 20. The word mole just means 6 times 10 to the 23rd. And it so happens that if you take 12 grams of carbon 12 and, exactly 12 grams of carbon 12, and you count the atoms in it, it's 6 times 10 to the 23rd. So, so it's just a counting number. So molar is just telling us how many numbers of molecules per liter and stuff like that. And there's a couple of different sort of abbreviations that are really handy when working with concentrations. So millimolar is 1 over 1,000, micromolar is 1 over 1,000,000, and nanomolar is 1 over 1 billion. And this is written out here. And once again, this idea, when you see this negative number as an exponent and whatnot, you can just read that as over. Okay. So we know we've got our critical concentration, and what we want to do is divide K off by K on. So K stands for the constant, the off constant, and the on constant. And we can sort of expand that. We know what our off rates and our on rates are. So, so to kind of expand things, so one can write the off rate like that, and you can also write things like this. And whenever you're working in the lab and you need to worry or work out concentrations and stuff like that, what I would suggest is getting out a piece of paper and treating it like a chemistry problem you would in class or whatnot. So you're gonna write things out. You wanna write out all of your units. So this is 1.4 over seconds, and then we have our 12 per micromolar per second written like that. Because our on rate is on the bottom, we get to flip that. And then you cross out the seconds and you're left with a critical concentration. So the sort of beauty of all of this is we can measure the on rates, we can measure the off rates, both as a function, well, first as a function of concentration. You put those together and you can get the critical concentration. Another way to think about this is you can just take a solution of actin, let it go to steady state, measure the critical concentration, and then you can use this number to also back out on and off constants, or to limit things or get you in the right ballpark and stuff like that. So when you first look at this and you go like, wow, this is really crazy and complicated, in some ways it's very simple, and it all just sort of folds into itself, and you get this simplicity where there's all of these different numbers, you understand how it fits together, and it tells you at the plus end of an actin filament, the critical concentration is 0.12. All right, so let's see. So, given this information below, what is the critical concentration? So go ahead and work it out. And I think you can do it in your head, probably. It's A, okay. All right. So yes, okay. And that's really, it doesn't need to be more complicated than that, so, okay. So in terms of the actin kinetics, so actin assembles when the free concentration is greater than the critical concentration. It disassembles when the free concentration is less. And one thing to note, the total critical concentration for the actin filament is going to be in between the plus and the minus ends. So if you think of it this way, is if we had a concentration of 1 micromolar, so is 1 micromolar greater than 0.6? There would be assembly on the minus end. And there would definitely be assembly on the plus end as well. So if we had a concentration of 0.1 micromolar, would there be assembly or disassembly on the plus end? Disassembly, and then at the minus ends? Of course. So for the filament as a whole, the critical concentration is going to be between these two numbers. I don't think it's the exact average. There's some other things that get weighted in there and stuff like that, but that's where the critical concentration is going to be found. So, and then, so I'll throw this up as a clicker. So we'll just set up some ideas. Okay, so we know the critical concentration at the minus end is 0.6. At the plus end, it's 0.12. And we know that we have a solution where it's 0.5 micromolar to begin with. So is 0.5 more or less than 0.6? Less, okay. Is 0.5 more or less than 0.12? Okay.
Oops, okay. I didn't read that right. That's good.
So we have a situation where the actual concentration is higher than 0.1 and it's lower than 0.6. So, given that analysis, let's look through the answers and we'll choose here again. So, do you have an answer in mind? Okay, and the answer would be
B.
B. Okay. And that makes sense. So yeah, so there's going to be polymerization at the plus end because this is greater than that, and then the other way around. All right. So if we go to 1 micromolar, are we good with A? All right.
Yeah.
Okay. Sydney, you're good? Okay, all right. And then at 0.1,
D.
exactly, yeah, okay. Why do I have it?
The first one too, you had D
Oh.
italicized as well.
Okay. I need to update these, so, okay. So here's an overview of actin assembly. So these are the subunits, the little yellow triangles are ATP. We get the nucleation event. There's a plus end with the back. The clef is on the minus end. After nucleation happens, initially there's going to be elongation at both ends. So we've got our plus end or minus. You can see assembly's faster on the plus than the minus. At steady state, the rate that monomers are added and lost are going to be equalized. One thing I don't like about this movie is it loses some of the subtleties of actin dynamics because what it's just showing you is that. But in many ways, this is a nice little movie to illustrate a few of the concepts, but the details of how assembly is going on at the plus and the minus ends are a little bit oversimplified. So, so with that, thank you all so much for being attentive and interactive and all of that good stuff. But yes, go ahead.
I have a question. Since the rate constants for the off and on rates are per second, does that mean like this assembly follows like a recursive decay?
Exactly, yeah. So the question is, so since the off rates are per second, does disassembly radioactive? Not quite in that way and stuff like that, but yeah.
Half-life after.
Yeah, you can definitely think of, if you had X filament, you could measure decay curves and stuff like that. But because the complication there is over time you would lose filament ends and then the rate of decay is going to actually slow down and stuff like that. So there's layers of subtlety under all of this and stuff like this, but this is, yeah, just a good way to start.